Look closely at this medical drip tube. In emergency rooms across the world, a severely malnourished, disoriented patient is brought in, their pulse fading. The medical team operates on a universally accepted instinct. A starving body requires immediate energy. They hook up a high-density bag of intravenous glucose, and the sugar enters the patient's bloodstream. Instead of stabilizing, the patient's biological systems violently collapse. Within hours, they descend into irreversible brainstem tissue death, slip into a coma, and sustain permanent neurological damage. The life-saving protocol directly triggers the exact condition it was attempting to treat. We can trace the root of that paradox to a completely different clinical setting. This glass test tube contains a blood sample drawn from that exact patient, resting on a laboratory counter. Sitting idle for 20 minutes under room temperature light, the blood inside this tube is actively degrading. Crucial molecules are cleaving apart. By the time the laboratory machine processes the sample, the evidence of the patient's true condition has chemically destroyed itself, effectively blinding the physician to the approaching collapse. The emergency room catastrophe and the degrading blood sample share a single point of failure: vitamin B1, or thiamine. This molecule acts as the thermodynamic grounding wire for human bioenergetics, determining whether a cell safely extracts energy or burns itself out. Inside human cells, this molecule exists in its active biological form, thiamine pyrophosphate, or TPP. Biologists identify it as exceptionally conserved. The atomic machinery driving TPP operates identically in primitive single-celled yeast and in the vastly complex biological architecture of humans. Evolution retained this exact structure to solve a massive thermodynamic danger. Human cells operate at a normal body temperature of 37 degrees Celsius. At that temperature, breaking stable carbon bonds to extract energy from sugar releases wild, unpaired electrons. If left uncontrolled, these electrons act as microscopic shrapnel, causing homolytic free radical damage that easily shreds DNA, proteins, and lipid membranes. This diagram displays the solution. Between the nitrogen and sulfur atoms on the thiazole ring sits the C2 reactive carbon node. This acidic node acts as an electron sink, absorbing volatile energy from broken carbon bonds to stabilize dangerous electrons. As a metabolic bridge, TPP links sugar breakdown to cellular respiration, serving as the mandatory coenzyme for pyruvate dehydrogenase. Without that specific C2 node grounding the electrical variance, human cells would literally fry their own biological machinery from the inside out every single time they attempted to extract usable energy from a meal. Given its critical role in thermodynamic stability, you might expect human physiology to maintain massive reserves of thiamine. It does not. A healthy adult possesses a total body storage of roughly 25 to 30 milligrams. This anatomical heat map reveals the body's evolutionary priorities. 60% is locked inside skeletal muscle for physical survival. 30% concentrates in high-demand organs, the heart, kidneys, and brain. In the bloodstream, the transport phase is tiny. Over 90% of circulating thiamine hides securely inside red blood cells, armored to prevent premature kidney filtration. The consequence of this fragile vault is a rapidly ticking clock. Human cells completely lack the genetic machinery to synthesize thiamine internally. Total starvation depletes the entire bodily supply in just 10 to 14 days. Certain physiological conditions drain this vault even faster. Pregnant women suffering from persistent, intractable vomiting lose their ability to retain nutrients while simultaneously trying to fuel fetal development, emptying their thiamine reserves in a matter of days. By discarding the biological hardware to build this molecule from scratch, our evolutionary ancestors permanently tethered human survival to the external environment. We are continuously locked 2 weeks away from total system collapse. In a starving patient, infusing high-density glucose triggers an immediate insulin surge. Cells frantically pull in sugar, hyperactivating glycolysis and generating a massive wave of pyruvate. That pyruvate accelerates toward the mitochondria, but the thiamine vault is empty. The pyruvate dehydrogenase gateway is blocked. The chemical flow hits a brick wall, backing up instantly. Unable to safely burn fuel, the cell shunts the backed-up pyruvate into a malignant lactic acidosis storm, instantly consuming any trace thiamine left in the tissues. This MRI illustration shows the localized destruction of that acid storm. Central bioenergetic failure shuts down ATP-dependent ion pumps in the brain. Calcium violently floods the neurons, causing symmetrical petechial necrosis, rapid cell death, and microbleeding, specifically targeting the mammillary bodies. Clinicians recognize this structural meltdown as the Wernicke triad. The patient exhibits erratic eye paralysis, ataxia leaving them unable to walk, and profound, listless global confusion. If untreated, this damage consolidates into Korsakoff psychosis. The physical lesions permanently destroy the brain's ability to form new memories. To cope with gaping holes in its timeline, the brain invents elaborate, completely false stories, a phenomenon known as confabulation, which the patient fully believes. Lacking one microscopic molecule for a few weeks physically dismantles the neural hardware required to record reality, replacing the patient's baseline reality with permanent hallucination. To avoid triggering that specific brain death, clinical pathologists must accurately measure the empty vault before a physician ever administers glucose. Ordering a standard serum thiamine test yields practically useless data. It only measures the transient fraction of the vitamin floating outside red blood cells, reflecting a recent meal rather than the true biological baseline. This cross-section illustrates the severe pre-analytical trap awaiting a whole blood test. Inside red blood cells, active TPP coenzymes share space with dormant phosphatase enzymes. If the blood tube remains at room temperature, those phosphatase enzymes activate and catalyze a degradation reaction. They strip the pyrophosphate tail from the TPP molecule, converting the active coenzyme into an unmeasurable transport form. By the time the sample enters the analyzer, the active molecules have destroyed themselves. The laboratory reports a falsely depressed thiamine level, severely misguiding the diagnostic process. Our advanced million-dollar diagnostic infrastructure can be entirely neutralized by a single tube of blood sitting unchecked on a desk. The evidence is extraordinarily fragile. The true gold standard for measuring the vault requires breaking open the red blood cell armor and evaluating the contents using liquid chromatography tandem mass spectrometry, or LC-MS-MS. This chromatogram dashboard zeroes in on the absolute chemical truth. The mass spectrometer filters out biological noise, isolating the active coenzyme's exact mass-to-charge ratio down to the nanomolar threshold. The instrument also bypasses the opposite clinical trap, mechanical hemolysis. Forcefully mishandling a blood draw ruptures red cells, leaking massive amounts of TPP into the plasma. This causes an artifactual elevation that can completely mask a severe, life-threatening deficiency. Modern hospital informatics platforms now integrate augmented intelligence to monitor these variables automatically. Algorithms cross-reference whole blood TPP with lactate and hematocrit data to generate a real-time cerebral bioenergetic vulnerability score. Diagnostic stewardship means mathematically spotting the smoke before the fire, catching the vault's collapse days before a clinician ever touches an IV bag. The physiological constraints of the thiamine vault establish the absolute law of metabolic medicine. In a malnourished patient, thiamine must always precede or accompany glucose entry. This syringe represents the only intervention capable of halting the destruction once Wernicke's triad begins. The medical team is fighting a ticking clock. A massive dose of intravenous thiamine must be administered immediately. Within hours of the first symptoms, this load can reverse ocular paralysis and stop the necrotic brainstem damage in its tracks. Delaying therapy past the 24-hour mark closes that window. The necrotic lesions finalize, permanently locking the patient into Korsakoff psychosis. The clinical record shows a chain of dependency. The C2 node enables the Pyruvate dehydrogenase gate. The 30 mg vault dictates the 2-week depletion timeline, and mass spectrometry provides the only accurate view into this fragile energetic vault. Recognizing these constraints allows hospital systems to enforce clear diagnostic protocols, acting to logically eliminate highly flawed serum testing from standard care orders. We engineer sprawling cities, decode our own genomes, and map the cosmos, yet our baseline reality remains entirely subject to strict chemical laws. Our highest cognitive functions, our memories, and our mechanical cardiovascular stability rest completely on a fragile, outsourced microscopic grounding wire that our bodies cannot even manufacture.